Hi everyone, it's Yiping here. Today I want to make a video about gene regulation in eukaryotic cells. The first thing I think is important to know is that eukaryotic gene expression is regulated at many stages, as you can see in this picture. So in this video, I'll focus on four major gene regulations happening in eukaryotic cells, which are chromatin modification, transcription initiation, post-transcriptional regulation, and post-translational regulation. So the first step, the first opportunity for cell to regulate the gene expression is the modification of chromatin structures. As the textbook says, chromatin modifying enzymes provide initial control of gene expression by making a region of DNA either more or less able to bind the transcription machinery. So the histone acetylation is an example that chromatin modifying enzymes make DNA molecule being more able to bind with the transcription machinery. In this picture, the green spots here represent the acetyl group, and this structure sticking out of the nucleotide represent the histone tails. So when the acetyl groups bind with the histone tails, it will make the structure of the chromatin being less condensed, so the components of transcription machinery will be easier to access the DNA molecule so that the transcription can be initiated. And the DNA methylation just doing the opposite things as the acetylation, because the addition of methyl groups to the histone tails can promote the condensation of chromatin. And the next phase that gene expression can be regulated is transcription initiation. The regulation of transcription initiation involves proteins that bind to DNA and either facilitate or inhibit binding of RNA polymerase. So before I talk about the regulation of transcription initiation, let me first introduce two key components of DNA structures of eukaryotic cells. This is a DNA template strand, and the green sections here is a sequence of DNA called promoter, and it is where the RNA polymerase will bind to. And the three yellow sections here represent control elements, and groups of them compose enhancer. There are two kinds of transcription factors that engage in transcription initiation. The first one is general transcription factors. Those are the proteins that usually bind with proteins such as RNA polymerase. RNA polymerase then bind with a promoter of the gene, and as it moves along the DNA strand, the transcription is initiated. But this kind of initiation has low rate of initiation and only produces a few RNA transcripts. The second kind of transcription factors are specific transcription factors. And it includes activators and repressor. Those are the transcription factors that have a high level of transcription. And this is an example of how activator works. First of all, the activator protein will bind with enhancer, and then a DNA binding protein here will bend the DNA to bring the activator closer to the promoter. After that, the activator will bind to certain mediator proteins and general transcription factors to form a transcription initiation complex. Now let's look at how repressor work to inhibit transcription initiation. Basically, transcription repressor works in these three ways. The first one is they bind directly to control element DNA, and the second one is they block activator binding, or in some cases turn off transcription even when activators are bound. And the last way is that other repressors block the binding of activators to proteins that allows the activator to bind to DNA. Another thing that I want to say about the regulation of transcription initiation is a cell type specific transcription. For example, in the nucleus of a liver cell and lens cell, they both have the genes for making albumin and crystalline are both present, but only albumin is present in liver cell, and only crystalline is present in lens cell, and that is because only the activators for albumin gene are available in liver cell, and only the activators for crystalline gene are available in lens cell. So the presence of some specific activators plays an important role in determining the function of the cells. And the next step that will regulate gene expression is the post-transcriptional regulation. This process involves two important parts in it. The first one is RNA processing. During this process, different mRNA molecules are produced from the same primary transcript, depending on which RNA segments are treated as exon and which are treated as introns. So, for example, in this primary RNA transcripts, if the gene 3 is treated as exon and 4 is treated as intron, it will produce this kind of mRNA. And if 3 is treated as intron and 4 is treated as exon, it will produce this kind of mRNA. And the next part of post-transcriptional regulation is RNA degradation. Nucleotide sequence that during the process of RNA splicing, the cap and tails will be added in the DNA molecule, and the untranslated region at the 3' end of the molecule here will affect how long an mRNA remains intact. And the last step for cell to regulate gene expression is the post-translational regulation. It also involves two parts, which are protein processing and protein degradation. For protein processing, we need to know that regulatory proteins are commonly activated or inactivated by the reversible addition of phosphate groups, and regulation might occur at any of the steps involved in modifying and transporting a protein. And for protein degradation, to mark a particular protein for destruction, the cell commonly attached molecules of small protein called ubiquitin to the protein, and the giant protein complexes called proteasomes then recognize the tagged protein and degrade them. So this is the process of protein degradation. The last thing I want to say about gene expression is that for some of these gene regulation is happened inside of the nucleus, such as the modification of chromatin, the transcription initiation, RNA processing, and the rest of them, such as degradation of mRNA, translation initiation, and post-translational regulation is happened in the cytoplasm. Yeah, that's all I want to say about gene regulation. Thanks for watching. See you next time.